Geoff. Let's take a look at Parkinson's disease. Neurodegenerative diseases were first described at the start of the Industrial Revolution. It's immediately apparent that the development of Parkinson's disease was caused by something that first occurred at the start of the Industrial Revolution. Let's consider some of the changes that occurred at the start of the Industrial Revolution. The first one was the consumption and commercialisation of milk. We would imagine milk was highly contaminated, not pasteurised, carried contagions, and was adulterated with metals like sugar of lead. Bread, like milk, was also commercialised and also adulterated with metals like aluminium. The Industrial Revolution is synonymous with heavy metal pollution, not only in the food chain but also in the general environment, with examples being coal replacing wood in home fires and resulting in the release of cadmium. All these heavy metals, lead, aluminium, cadmium and arsenic, have all been linked with Parkinson's disease and other neurological disease. It is unlikely, however, that heavy metals alone would be the cause of Parkinson's disease, as most of the population is similarly exposed to these heavy metals, and yet most of the population don't get neurological disease. It's also the case that many other substances like pesticides have been linked with Parkinson's disease, and they never existed in the 1800s. This suggests that heavy metals may play a part in Parkinson's disease along with pesticides. Do they have something in common, or do they play a part in the development of Parkinson's disease? Surprisingly, heavy metals and some pesticides do have something in common. So let's have a look at that. On this table, we can see that those elements and those substances that are most closely linked with the development of Parkinson's disease are actually the ones that have a zero hydrogen bond count, a zero hydrogen bond acceptor count, a zero rotatable bond count, and they have no function in the human body. Detective Geoff calls these substances zero bond gang, and they can consist of traditional heavy metals, or they can be synthetic chemicals that are recently made. Therefore, we have a continuity of the potential to cause Parkinson's disease developing first from the Industrial Revolution. When we first begin to study Parkinson's disease, we find that there have been many associations between possible causes and Parkinson's disease. However, for things to advance beyond associations, we need to develop an understanding of possible mechanisms. To do this, in this case, let's have a look at how zero hydrogen bonds work. Simplified way, substances are formed from molecules, and molecules are formed when two or more atoms bond together through chemical interactions to achieve stability, usually by filling their outer electron shells. These bonds are created when atoms share electrons, they are covalent bonds, or transfer them in ionic bonds, creating new stable units with properties distinct from their original atoms. In this sense, any substances that has a zero hydrogen bond has an impact upon how atoms and molecules are formed. Therefore, it is a significant part of the chemistry and the construction of molecules. The heavy metals and zero bond count substances, by our definition from the previous slide, clearly mean that these substances are not liable to form bonds with oxygen or nitrogen, that they are not able to donate atoms to other structures to form bonds, that they are not flexible. These zero bond substances can be chelated by sulfur bonds, cysteine, and some specific proteins. There is no dedicated transport and metabolism process for zero bond substances that have no function or use in the human body. This means that these substances are intervening in proton formation and creating delinquent molecules like we find with alpha-synuclein or tau protein dysregulation. They also have the potential to interrupt the formation of pink-like proteins. On these substances, or equally, these substances are hijacking the essential proteins and depriving their availability to be used in the formation of proteins. Detective Geoff says that the zero bond gang are working in partnership with something else. Let's go back to milk. We assume milk no longer contains impurities like heavy metals that it once did. So for it to still be involved in neurodegenerative disease, there is another component of milk that binds the zero bond gang and transports them across the blood-brain barrier. That element is triglycerides. Triglycerides are the dominant constituents of milk fat, 98.3%. Detective Geoff decides to look into the character of triglycerides. Triglycerides are not able to be directly absorbed in the human digestive system. Instead, they are first broken down into mono and diglycerides and then absorbed across the small intestine and then reformed back into triglycerides to be transported around the body. This creates an interesting question. What happens if something something goes wrong in the reforming process and the body is exposed to higher levels of mono and diglycerides. In this recent study, it was established that mono and diglycerides were first identified in the human brain and that a clear association of increased levels of mono and diglycerides was seen in Alzheimer's disease patients. The conclusion was that a mono and diglyceride metabolic defect was a central correlate of chemical and pathological progression of Alzheimer's disease. This study confirmed that glycerides have been found in synuclein Lewy bodies and that glycerides may contribute to Parkinson's disease. Mono and diglycerides are used as emulsifiers in food products as they bind to heavy metals and help stabilize food. So for the first time, we see that glycerides and heavy metals, or the zero hydrogen bond gang, may well be working together to create metabolic dysfunction. Environmental metal exposure is associated with elevated triglyceride levels. It is established that the two elements have a symbiotic relationship. Here are some more specific examples of metal triglyceride interactions. Detective Geoff draws the following conclusions. Lipids and triglycerides have been associated with neurodegenerative disease. That glycerides are converted to monoglycerides to get through the gut wall and are then converted back to triglycerides, and that there appears to be little, if any, studies on what happens if monoglycerides are not converted back. That mono, di, and triglycerides will attach themselves to heavy metals, and this is a result of the zero bond nature of heavy metals. That by direct implication, those people exposed to high triglyceride consumption at the same time as exposure to the zero bond gang will be at greater risk of neurodegenerative disease. Detective Geoff then considers if there is an example where humans are exposed to mono, di, and triglycerides at the same time as the zero bond gang. The emulsifier E471 is a monodiglyceride substance, and Detective Geoff knew from personal experience that it was capable of causing neurological disturbance. Some foods have hidden E471. However, the overall percentage of all foods that contain E471 is 7%. The subcategory ice cream has 75% of products containing E471. In a European study, over 33,000 products listed E471 as an ingredient. E471 should be mono and diglycerides of fatty acids. So Detective Geoff had some samples tested for heavy metals. This is what he found. Three important aspects of what Detective Geoff found are: 1. The variation of what is in E471 is considerable and no consistency exists. We didn't test for other chemicals like pesticides and PFAS or microplastics. 2. All the substances identified have a zero hydrogen bond donor count, zero hydrogen acceptor count, and zero rotatable bond count. 3. Interestingly, the Australian sample was the only sample that arrived in aluminium foil packaging. This suggests that even without any encouragement, the E471 bonded to the aluminium from the packaging. A sample diet for one day could result in 4.37 grams of E471 consumed per day. This demonstrates that each manufacturer will say, well, we only use such a small amount of E471 that it wouldn't affect anyone. But they never discuss the cumulative effect of E471. Considered E471 in the last slide, but now we know that E471 comes with heavy metals attached. E471 carries a double punch. It's worth keeping in mind some general principles in regards to heavy metals. Heavy metals that have no biological function build up in human brains and are difficult to remove. The standards and the World Health Organization do not recognize this when establishing safe levels. Long-term neurological effects are not considered when considering the safety of a product. The heavy metal burden, and in fact all substances that have the zero hydrogen bond structures, multiply the burden and build up in the brain. These substances all have the ability to interfere with the metabolism and function of proteins. This means that neurological disease may not be the result of one substance, but in fact multiple, and the combinations could determine the type of and degree of neurological disease. The cumulative effect of all the toxins could well cause the response mechanisms to become overloaded and result in reactive oxygen species type damage. Some of the other heavy metals are reported to have up to 27-year lifespan in the brain. This creates a scenario where even the smallest amount over years just keeps adding to the burden. So in E471 we have a real Frankenstein, in that there are three ways in which it may be involved in neurodegenerative disease. Firstly, the primary ingredient, the glycerols, are capable of disrupting protein formation and function. In the second way, the impurities in the E471 in the heavy metals can cause neurodegenerative disease, and in the third way, the glycerols can act as a transport mechanism for the heavy metal toxins to reach the brain. Accumulating evidence underlies a significant role of lipids in the pathological pathways leading to Parkinson's disease. Besides the well-described lipid alteration in idiopathic Parkinson's disease, This review summarises the several lipid alterations observed in experimental models expressing Parkinson's disease-related genes and suggests a possible scenario in relationship to the molecular mechanisms of neural toxicity. Parkinson's disease could be considered a lipid-induced proteinopathy, where alteration in lipid composition or metabolism could induce protein alteration, for instance, alpha-synuclein accumulation, and finally neuronal death. Lipid studies and their role in Parkinson's disease is a rapidly growing field. From our previous studies, we know that zero hydrogen bond substances are attracted to and bind to mono and diglycerides, and we now find out that alpha-synuclein also has a preferential binding to the lipids. We also suspected that when zero hydrogen bond substances bind to the lipids, they will alter the ionic charge. This may then cause the lipid to mimic cardiolipin. However, the addition of the zero gang member is at least going to result in dysregulation. Research has shown that mitochondrial metabolism is clearly influenced by the lipids and their levels. The lipid environment within mitochondrial membranes influence all of these processes as they determine membrane curvature and structure, as well as affecting the recruitment and activity of specific proteins. The role of dysfunctional mitochondria in the pathogenesis of Parkinson's disease has been demonstrated in several models, including aging yeast, as well as in the Parkin-deficient transgenic mouse models. Dysfunction in diglyceride kinases have been shown to be involved in Parkinson's disease pathology. Monogenic familial Parkinson's disease is caused by mutations in 6 genes: SNCA and LRRK2, Parkin, DA1, PINK1, and ATP13A2. These studies suggest that the interaction of alpha-synuclein with lipids is physiologically important and that PD-associated mutations may impair the normal function of the protein. For a more detailed explanation, please read the next 3 slides at your leisure. Lipidomic studies have displayed an increasing level of connection between the role of glycerides and the development of Parkinson's disease. Even in early onset or genetic PD, it has been shown that genetic alterations are directly involved in the development of lipid dysregulation or disturbance. 2 aspects have yet to appear in the research. They are: 1, there is no information on a role for monoglycerides. This could be significant for both tri- and diglycerides are linked with Parkinson's disease. 2, the existing research has only started to consider the role of glycerides, However, in this study, I have identified that zero hydrogen bond substances, mainly heavy metals, have an affinity for glycerides, and in substances like E471, they represent a perfect model of how diglycerides could be altered by the zero bond gang to change the charge of glyceride and interfere with the function of proteins like alpha-synuclein. Detective Geoff identifies the offenders. Offender 1, the zero bond gang. This gang have existed forever, but they have extended their gang by including synthetic chemicals. They have no place or purpose in the human body, and because of their zero bond characteristics, they interfere with protein structures and are very hard to remove. Offender 2, the glycerides are capable of causing misformed proteins like alpha-synuclein, but they also act as the transporters of the zero bond gang. More is known about triglycerides, but it is the mono and diglycerides that present the greater risk as they are designed to pass through human defense systems. Glycerides only come into wide use with the introduction of milk bread and now substances like E471, which represents the perfect substance to cause neurodegenerative disease. Feel free to read this slide if you're interested on Detective Geoff's methodology. Detective Geoff then wanted to test some of this theory. He wanted to see if differences existed between himself as a non-Parkinson's person and his mum, who has Parkinson's disease, and so he conducted a hair test. This is what he found. Detective Geoff found that his mum had a very highly dysregulated lipid function, whereas his was normal. He also found that his mum had greater susceptibility to lead and cadmium and rare earths. He thought maybe rare earths could be the next risk for Parkinson's disease. White matter disease is generally considered a part of aging. However, with the close correlation of symptoms with Parkinson's disease, the question is, does Parkinson's disease result in white matter disease, or are both connected with lipid dysfunction? And a final thought. Almost every day we read of new connections and proposed mechanisms for the development of Parkinson's disease. However, with each new angle, it comes back to the central issue of lipid dysregulation.